Hi everyone, welcome back to the channel. This is Dr. Asi and this is Dr. Asi Explains. So today we are going to actually do a review on 3 chapters for final exams, which is coming up. So the chapters are 12, 13, and 15 from your green book. Chapter 12 is Mendelian Genetics. Chapter 13 is Chromosomes and Chromosomal Theory. And chapter 15 is Gene Expression, Gene to Protein. So let's get started. Okay, so we start with chromosomes. This is again chapter 13, very important chapter. So the chromosomal theory. So everything starts with chromosomes and DNA in the chromosomes, and then genes in DNA, and then all these 3 chapters are linked to each other through these chromosomes, right? So this chapter talks about a lot of experiments done at Columbia University by Dr. Morgan, basically, and he's receiving a Nobel Prize because of his work and his development of chromosomal basis of inheritance. And his model organism was fruit flies, Drosophila and Melagaster. And Dr. Morgan showed that Mendelian inheritance and its physical basis in the behavior of the chromosome. So he took the Mendelian genetics, he moved it to the physical basis in chromosomes. So you can think of the chromosomes as like highways, and you see here in this picture, basically in human genome map. So you can see here, this is a male person, which actually is from largest chromosomes, number 1 to the smallest number Y chromosome here. So, and all the known major genes actually in each chromosome that code, they are actually marked in this chromosomal map. This is all started with actually Dr. Morgan's experiments in 1904 at Columbia. So he specifically worked on some genes. They are actually very interesting because they are not in regular chromosomes or autosomes, but they are really on X chromosome specifically, or Y chromosome. He called them sex chromosomes. Basically, they exhibit unique patterns of inheritance than different than Mendelian genetics. We learn. And then we talked about, of course, another very important topic is linked genes. So linked genes are, you see here, 23 chromosomes, basically 22 plus XX or XY, and basically each chromosome has many genes, maybe 1,000 genes on them, right? So, so if 2 genes are on same chromosome, like chromosome number 1, you see here in this picture, they are called linked genes. They are linked to each other, right? So they are linked genes, and also they don't follow Mendelian rules. And also, lastly, alteration of chromosomes. If what happens in some genetic disorders that caused by alteration of chromosomes and some inheritance patterns that they don't necessarily follow Mendel genetics. So this chromosomes, again, that's where genes are located, and then they could be linked or unlinked. If they are linked, they again, they are so close together that they move together. If they are unlinked, like most of the genes that are actually studied by Mendel, then actually they go through independent assortment, and so they actually they behave independently. Chromosome can be autosomes, just 1 to 22, or sex chromosomes like XX is female, XY is male, and they can carry, these sex chromosomes can carry X-linked or Y-linked traits, different than besides their determination of gender of next generation. So and you see here, this is a map. This is a chromosomal map, and this map is actually is like highways. You see here, and then each chromosome is like one single linkage here, and then there are specific distances there between the genes in the map. So 1% So they are named one map unit, which means 1% recombination frequency. And so that we can map the location of the gene. So next step will be cloning the gene based on this location of that specific gene. So make sure you go over each of these and study these specifics in more detail in chapter 13 in chromosomal inheritance in our biology 2E book. Okay, so the next is well, Mendelian genetics. We talked about a lot about Mendel. So the Mendel is the basically considered father of genetics and came up with the gene idea. As you see here, his plants. He was a plant biologist also, and used this scientific approach to identify two laws of inheritance, probability laws, and then inheritance patterns, often more complex than maybe predicted, and then many human traits. Today, we know they don't necessarily follow Mendelian genetics. So Mendel Genetics, obviously, is in our book is chapter 12, which is basically, again, chapter 12 from the book. If you look at in study, you should always start with the basics vocabulary of Mendel Genetics, which are what is genotype, what is phenotype. Always remember phenotype equals genotype times environment. Phenotype can be wild type or mutant. Genotype can be homozygous, heterozygous. We name the genes with a letter from the alphabet. If it is big letter, that means dominant. That's different than little letter, which means recessive. And then dominance can be just complete dominant or codominant, like we see in blood types, like A, B, and then incomplete dominance, like we see in some plants like snapdragon. So make sure that you study these specific differences, and then make sure you know the words and their roots. Co means together, di means two. So Mendel studied mono and dihybrid. That means one gene and two gene problems, and he came up with 3:1, 9:3:1 ratios. Make sure that you actually go over this. Epi means besides. Epistasis means standing besides, a type of interaction between two genes, one gene actually affecting the expression of second gene. Heterozygous means different, like big A, little a. Homozygous means same, big A, or little a. Pheno means appearance. And poly means many, like polygenic, like we see in some cases. So make sure you know the definitions of gene, allele, which is basically different version of the same gene. If the gene is eye color, and then allele will be blue eye color, green eye color, brown eye color, hazel eye color. And these are all called traits: eye color, dominant, recessive, genotype, phenotype, homozygous, heterozygous, test cross, and monohybrid cross or dihybrid cross. And then make sure you know some specifics. Again, look at the book and do more reading on these chapters, right? So this is in the book, chapter 12, Mendelian genetics. And last chapter will be on the final exam is chapter 15, which is gene to protein. It's really how genes work, gene expression, which is a combination of transcription and translation, right? So genes, now we know genes, but genes has to specify proteins following transcription and translation to processes. That's how they work. This chapter is about how genes work. Transcription is the first thing, DNA directed synthesis of RNA, but specifically mRNA in this case. And eukaryotic cells have to modify this RNA after the transcription. Translation is the second big process, which is RNA directed, and then synthesis of polypeptide, which makes the proteins eventually. Mutations can happen because of one or few nucleotides can affect the protein structure and function of the final polypeptide. So gene-specified proteins, again, via transcription and translation. So make sure you actually really study this. Look at this whole model that we have actually in the book, how actually they are following this, what structure, right? So anti means opposite, exo means out, intra means intra, within, poly means many. You already seen that. So go over this, and then this is chapter actually 15. Make sure that in our green book, chapter 15, biology 2E, and then make sure that you actually you know how genes work. Combine it with the other two chapters, Mendelian genetics and chromosomes, and and then if you have time, do more review on all these three chapters. And now we have more time even for the final exams. So best of success in the final exam. Thank you for watching, and best of luck. And don't forget to subscribe to our channel. See you in the next video.